So, uh, at this stage, we can move on to the next section of the lecture, in which we'll talk about disease mutations that are associated with RAS. And I think now that you guys understand how growth factor signaling works, it's going to be a lot clearer how these disease mutations function. So, one thing that you're going to quickly discover about RAS, um, the more you read about it and the more you learn about its role in disease, is that every single step in the RAS catalytic cycle has disease associated mutations with it. And so we're going to go through each component, each contributor to the RAS catalytic cycle, the GTPase, the GEFs, the GAPs, the effectors, and go through examples of how mutations lead to utter catastrophe in this system that cause disease. And so, of course, the first one to consider are mutations in the RAS protein itself. And so, more than 30% of all human cancers, including 95% of pancreatic cancers, 45% of colorectal cancers, they are driven by direct mutation of the RAS family of genes itself. Think about that. If you know someone or you someone has pancreatic cancer, it is almost certain that the RAS GTPase protein here is mutated. And this means that if we could really understand how to fix these problems, we could make a huge impact on human health. So what are these mutations? Why are they causing problems? Why are they causing disease? Well, one of the most common mutations you'll hear about is this protein, is a position called glycine 12 in RAS. And so glycine 12 occurs in this loop region here that sits right over top where the nucleotide binds. And this glycine at position 12 creates the space that's needed for gap proteins to be able to insert that arginine finger that we discussed on Monday into the active site to stimulate hydrolysis. And glycine basically is like a blank amino acid. It's just a hydrogen. There's no methyl groups. There's nothing there. And so any other substitution for glycine in this position just blocks the insertion of this arginine finger into that slot because there's just not enough space for this arginine residue to get in there and do its job. And a second mutation that is extremely common in the RAS protein itself occurs at position Q61. And Q61, this glutamine residue, um, as you'll recall from the Monday lecture, is a catalytic residue that's involved in GTP hydrolysis by RAS. And cooperating with RAS gaps who stick their finger in here, this glutamine is needed in order for the GTPase to be able to hydrolyze the GTP. And so if you mutate this to something else, you're going to result in molecules that can't be turned off. And the inability to turn RAS off is going to result in sustained activation of RAS in the absence of growth factor. And what that means is cells are basically thinking, hey, this is when I should grow. I should be growing right now. This is the time to grow, even though nobody's told them that they should be growing right now. There's no growth factor out there saying time to grow. And this means you've got uncontrolled growth. And uncontrolled growth is one of the major contributors to cells adopting a cancerous or oncogenic state. Now, in addition to the RAS protein itself, GAFs, GAPs, effectors, and signaling receptors that activate RAS can also be mutated, and we're going to go through these now. So, RAS gaps are definitely one major contributor to disease state. So, mutations, for example, in the GAP NF1 are associated with the disease neurofibromatosis. So, this causes tumors to develop along the nervous system, which can grow anywhere on the body. Often, you'll see them first in the eyes of someone, so they have these small tumors in the retina here. And these are basically coming because a critical gap protein that's needed to turn off RAS in certain cell types has lost its activity. And so again, you're going to end up with too much activated RAS when you're not supposed to. In addition, mutations in RAF, so if we think about growth factor signaling, we know that one of the functions of RAF is or one of the functions of RAS signaling through growth factors is to activate RAF. And there are mutations in RAF that can bypass the requirement of it to need GTP bound RAS for its activation. And what this means is you've got um, proteins that are downstream of the RAS GTPase um, that can now bypass the need for any regulated inputs upstream. And so what this means is you're going to have kinase molecules that are essentially constitutively active, and they're going to go on and propagate signals telling cells to grow, which again can stimulate cancer progression. Now, mutations in GEFs, you might be like, okay, that's going to be a big one because GEFs turn on the GTPase. But in fact, it's less typical to see GEFs associated with cancer. But mutations in GEF do cause other more specific types of diseases, which are called RASopathies. And an example of this is that there are mutations in the son of sevenless GEF that are associated with developmental diseases. For example, this one called Noonan syndrome, in which when an individual grows throughout their life, instead of developing normally, they end up with a variety of uh, skeletal morphological defects um, that ultimately it's not only the appearances but the health and biology of this individual is severely compromised. And then finally, remember that GEFs, GAPs, and their activity are all regulated by receptors that set how much GEF activity is going to be present, how much GAP activity is going to be present, and so on and so forth. And so there are mutations in growth factor receptors that can render them constitutively active. And in these cases, what happens is a receptor that normally will not have phosphotyrosine uh, residues present unless growth factor is received will put those phosphotyrosine residues on all the time. And as you know, that phosphotyrosine is what's going to recruit your SOSGEF to the plasma membrane and trigger activation of RAS. And so these mutations end up driving ligand independent activation of your RAS GTPase. Um, And that again means that a cell that normally should be waiting to grow, proliferate, etc., until it receives an appropriate signal, is now just going to start doing that completely independent of those inputs. And again, that can lead to uncontrolled growth and eventually cancer. And one of the things you might be wondering is like, well, it's interesting that we went through that tree, or that we kind of went through all the examples, and sometimes the GTPase was really produced a strong phenotype, but the GEF didn't produce a cancerous phenotype, and not all GAP mutations are as deleterious as others. So why might that be the case? Well, one thing you should think about is that inside your body, you have many different cell types that have many different gene expression profiles. So a muscle cell is different from a neural cell, different from pancreatic cell, and so those different cells may have different usage of specific GEF gaps, or effectors that they use as part of their ordinary function. And this plot, while it's a little bit confusing, this is an example of each dot in this plot here corresponds to a cell in the human body, and the amount of one particular gap protein called P120 is shown on this axis. The amount of HRAS expression in that cell is shown on this axis, and the amount of RAF expression in that cell is shown on this axis. And the main point I just want you to see here is that there are cells that have very different uh, utilizations of these components. So there are some cells that have lots of P120 gap, and some cells that don't have very much at all. And what this means is that each one of these different cell types may respond very differently to perturbations, mutations in these components. And a direction the field is really trying to go is to understand why some of these mutations actually are more deleterious in these cell types but not others. And so, just as a really relevant example, I mentioned before that nearly 100% of pancreatic adenocarcinomas are mutated in KRAS signaling. Yet, if you found someone who had kidney cancer, less than 1%, almost none of those cancers are ever associated with RAS mutations. So if a kidney cell gets a RAS mutation, it doesn't seem like that's going to drive cancer progression. But if a pancreatic cell gets this KRAS mutation, it's like game over. Like you are in some serious crisis. And yet we have no idea why. Why is it so bad to get this mutation here versus there? It probably has something to do with those different expression profiles of the other components in the cell, but we just don't know. And if we could explain differences like this, then it could really help us develop better therapies for people with these diseases.